Hey everyone, it's great to see you. I'm excited. So we wanted to get back to educational things. So we figured we would need a good guinea pig to test to see if we can do the Zoom thing. And Dr. Seth Toback is a good friend of mine. He and I worked together at his last company, my current company, PMRU, and he decided that we have one of the first test in the U.S. for COVID. We make a couple different tests and from different scientific methods to test for COVID and to test for COVID antibodies. But Seth decided that wasn't hands-on enough for him to make a difference in this pandemic. So he left the testing environment to go back to pharma to actually head up clinical trials for a COVID-19 vaccine to be more involved in directly saving lives. So I thought it'd be great if he can come and talk to us a little bit about that vaccine development process. And this is like a pretty unique set of vaccines. And I figure we'll do a Q&A afterwards and we can hit him with every pseudoscience myth about vaccines and he can dispel them for us. So with that, I will turn it over to my good friend, Dr. Seth Toback.
Great. Well, thanks, Matt. It's a pleasure to be here. I enjoyed talking about COVID. I enjoyed talking about vaccines, and I also enjoy dispelling myths. I'm a big fan of vaccine efficacy, and I picked this title here especially for Todd. It's kind of sounds smarter than your friends after watching the TV news about COVID-19 vaccines. So with that, I'll jump right into it. So my first thing is I have to give my disclosures. I just thought, Dr. McCoy was probably the first doctor to be the classic great doctor at disclosing. But he wouldn't disclose in the classic way by saying, I have this problem, that problem. He would tell people what he doesn't have or what he isn't. Like damn it, Jim, I'm a doctor, not a forklift. So I would tell people that, my disclosures for Dr. McCoy are: I'm not a regulatory specialist. I'm not a manufacturing specialist. I'm not a specialist at public health. I am a specialist in vaccine development, a specialist in primary care and pediatrics. And my other disclosures are I work for Novavax as an employee, and all of the things that we're talking about today here are basically my own opinion and not the opinion of the company. And we're just going to talk about multiple vaccines. And I think our vaccine's in there too, because it's one of the many vaccines that has data available. And you'll get to see my running Star Trek joke in here because one of the big funding agencies of vaccine development in the U.S. is Operation Warp Speed, which is a great opportunity to throw in some Star Trek jokes if you had the opportunity. So I took that opportunity. So who am I? This is a picture of me. Those with a very keen eye. My notice in the top right-hand corner, there's some old timey writing. This was taken at the Hobbit movie set in New Zealand. And so I'm a pediatrician. I'm a vaccine advocate. I've been working in the field of vaccines, respiratory viruses, small molecule treatments, and diagnostics for the last 15 years. And here's our agenda. We're going to be talking a little bit about vaccine candidates, maybe a little bit about some vaccine technologies. We're going to talk a little bit about COVID trials and how they're done. It's really a potpourri of the COVID topics. We could talk about this for a month. I could talk about this for a month, but we'll just keep it down to this one hour segment. We could talk a little bit about the variants that are going around right now, and then hopefully leave plenty of time for Q&A. So let's talk fast. So where are we in general in the world of COVID vaccine development? There's a lot of players out there right now, which is a great sign. There's multiple vaccines that are in phase 3, and we've really marched those along. So some being approved, some being in phase 2, and a lot in phase 1. We'll talk about the phases in a little bit. But basically, right now, there are about 4 vaccines that are approved. There are 6 that are authorized. And what that means is approved technically means it's fully approved and licensed in that country. Authorized means it probably has an early approval. Like in the U.S., we call that EUA, emergency use authorization. Sometimes they call it a conditional approval, a conditional marketing approval. So some of them are conditionally approved. The ones that are mostly 100% approved are the ones that are in kind of nefarious, questionable places like China and Russia, where they basically just said, it's approved. They slammed the gavel down. That was it. We're not going to talk much about those because we don't really have great access to their data and can't really talk about them because we don't know how accurate or how good that data is. So we're going to talk a little bit about a handful of the leaders in these categories. We can't talk about all of them. Can't say about all the lovely 37 vaccine candidates in phase 1 still. but we can tell you about the ones that are leading the pack right now. These are the ones you see on TV. So who are approved or conditionally approved? So the ones on the left are the ones that you're going to see in the U.S. The ones on the right are the ones you're not going to see in the U.S. And we're not going to talk about those quite as much, but just to mention that you're not going to see them. So on the left, you're looking at Moderna, which is the small biotech out of Cambridge. The Pfizer BioNTech, which is they're affiliated with a smaller company in Germany. So Pfizer is a U.S.-based pharmaceutical company, and then AstraZeneca is working with Oxford. So those are the ones that currently have some sort of approval. The ones on the right have approval in their respective countries, but again, you won't see the Chinese or the Russian ones. I do call out with the asterisk here, the ones that are involved with warp speed, and that's a slide that I have, a couple of slides. I'll tell you a little bit more about that program. So who are the general leaders that are not quite approved yet, but are getting there, because you see them in the news as well. So adding to that list is Novavax and J&J. So Novavax and Johnson & Johnson or Janssen, which is the pharma arm of J&J, have been making some news. The other big warp speed player is Sanofi's GSK. So this is I work with the Sanofi vaccine with a GSK additive, and they are a big player. They just got a later start. They're in phase 2, but they will be a big player too. So I thought we'd mention them as well. And excuse me while I drink. I've been on Zoom calls since 7 a.m. So I talk way too much and start to lose my voice with these things. So Operation Warp Speed, you hear this a lot. What is exactly Operation Warp Speed? It's a government agency. It's, you can see down at the bottom left that there is the alphabet soup of the companies or the agencies that work with it. Health and Human Services, CDC, NIH, BARDA, which is the biodefense arm of the Department of Defense, and the DoD. So these are a whole bunch of government agencies, and they're really, their raison d'etre is to be able to bring vaccines and therapeutics to market by either supplying money or supplying other resources and supplying some brain power, and also cutting the red tape in government to make sure this thing happens quickly. And their basic goal is to bring 300 million doses of vaccine into the U.S. They even have their own logo, look at the bottom right. So they have the little shield. And I don't know if there's any Star Trek fans out there, but that logo reminds me a lot of the Borg cube. I don't know if that is their inspiration, but I thought, they like Star Trek. They got the Operation Warp Speed thing going. They picked the cube. It's got to be the Borg cube. I don't know, but you can tell me what you think. But I think so. The other thing I'll say about Operation Warp Speed is probably wasn't the best name to characterize a process where you're trying to instill some confidence that things weren't done too quickly. So although I will say the name is growing on me, but initially I thought it was a little bit too aggressive of a name. It's starting to grow on me a little bit now, but there you have it. So the vaccine technologies, what kind of vaccines are out there and how they're working and which ones are new and what's the story with them? So I'm going to give you a couple of little cute diagrams of the major technologies out here, and we're going to start with the ones that's the newest. So basically the mRNA or the DNA technology. And really what this is looking at, this is you have to think back to your high school days when you learned some biology. What this is doing really is taking we'll start with mRNA first because it's a little bit easier. The mRNA, which is the part of the nucleic acid that's directly used to make protein. So if you recall, the way proteins are made is you have your two strands of DNA, they peel apart, and then the mRNA comes over it and it makes almost like a mirror image of the DNA. And then this mRNA goes into the cell, it finds itself a nice happy ribosome, and then it uses that mechanism as a factory to pull the amino acids together and arrange the amino acids in order to make a protein. So that's how proteins are made. That's how we can make cells for our retinas and cells for our hair so we can see things. And what this vaccine does is it just basically takes the mRNA that codes for the spike protein on the virus, and it puts it into your body, and your body does what it does when it sees mRNA. It makes protein. So the spike protein, as you probably recall, when you see all those pictures of the coronavirus on TV, with the big red globules effect on it, those are the spike proteins. And those are the proteins that there are probably the predominant surface protein, probably the best target to create a vaccine candidate for. So it's a great place to mount an immune response and to target antibodies for it. So really, almost all the vaccine candidates are using the spike protein. So what they did is they took through the genetic sequencing of the virus. They took the sequence of the mRNA in the virus, and they took out the segment that coded for just the spike protein. And within 45 days, this is how fast it was. Moderna was able to take, when China first gave us the entire list of the sequence, within 45 days, they could make their first mRNA sequence to work as a vaccine candidate. So they can actually make these mRNA sequences quite quickly because they're not quite as complicated as making a complete protein. So again, the end unit, the end game here is to make some spike proteins. So they take the mRNA, they put it in a little fat blob, which is what you see. Using my little cursor here is what you see over here. It's a little fat blob, and this is helps it to enter the lipid-laden cell membrane. Gets inside the cell. Again, it finds that ribosome, a little factory for making proteins. It makes spike proteins. The proteins get extruded from the cell, like you see over here, and then they present themselves to a antigen presenting cell, which is how the immune response starts. So really, it's just making this spike protein yourself. You're the factory for making spike protein. The DNA viruses work the same way. So they just use DNA instead of mRNA, and they have to go through a slightly different process. The funny thing about the DNA vaccines is they need a special device to get them into your body, which is one of the downsides of DNA vaccines. And then one of the downsides of the mRNA vaccines, is the ultra low temperatures that are required for shipping and storing them. The other thing is this lipid membrane that has put it in tends to cause a little bit of pain and discomfort when it's injected into the body. So those are the nucleic acid type vaccines. Let's go on to two other commonly used vaccine types. Let's go on first on the left here, the viral vectors. So the viral vectors are basically the J&J and the AstraZeneca one. And what they do is actually take the same concept. So instead of taking the mRNA and putting it in a blob of fat, putting it in a vaccine, sticking it into your arm, it actually takes that mRNA and puts it into a virus. And the virus then does what viruses are good at, which are invading your cells. So they take a virus and basically they rip out all the guts of the virus and they put it in the guts that they want. So it's like stripping down your car engine and putting in your own engine. That's what they do. And then the engine acts like a Trojan horse. It can get into your cell. So they take the mRNA that, again, codes that spike protein. They put it in a very common virus that infects you and me, like adenovirus, which is a common cold virus, or the measles virus, which also does a really good job affecting cells. They put it in the vaccine. They put it in your arm. The membrane of the virus gets into your cell like it's supposed to do. The mRNA comes out, finds that ribosome, makes the spike protein, and you activate the immune system. So that's how the viral vector vaccines work. So again, pretty simple technology. Now the protein-based vaccines are also very simple too. They basically just take the protein that you're interested in and make it external to the body. So in the last two cases, you were the factory making the spike protein. In this case, they take another external factory, like
that was going to be for today, but we had the power outage for tomorrow.
Melinda, keep yourself on mute.
Sorry.
I'll have to find the right button and do it myself, which I don't want to do. So the protein-based ones is basically the protein is made externally. So in the case of our vaccine, the Novavax vaccine, we actually take a cell from a moth that's been immortalized, and we put the mRNA into that moth cell, and the moth cell cranks out the highly purified spike protein. Then we take it out, we remove all the moth protein extract and other benzines, any other chemicals in there, and purify that protein, put that in a vaccine, and we give it to you. So again, it's a very common way to do it. The vaccines that we use, I call it the best of the old technology and the best of the new technology. protein-based vaccines have been used for decades. Think of the flu vaccine. Flu vaccine has been around since the 1940s, but they do it in a very old technology. They actually take an egg, they put in some virus, and they let the virus grow in the egg, and then they pull it out and they try and clean up all the egg. In our case, we actually take the mRNA and we put it into a insect cell and have the insect cell do it. So it's done with a much more sophisticated technology. And then the other thing that we do at Novavax is we then arrange that spike protein into a three-dimensional nanoparticle, which increases its ability to kind of show off the business end of the protein to create an immune response. And then the last category is probably the oldest. You know, back when they used to take, think back about things like smallpox. The very first version of smallpox was basically just a weakened version of smallpox. It was cowpox. They had this virus that looked a lot like smallpox, but it affected cows. It was a weakened version. They would take the whole virus and stick it in your body, and then you would get immune to smallpox. Some other technology we use would actually take another virus and just kill it. And you take the virus and heat it up, they take the virus and put it in formaldehyde, and they take the whole virus and they put that into the vaccine, stick it in your arm and give it to you. And that's done in a lot of different other routine vaccines you see in the market too. These ones right now are probably the slower ones that come into fruition right now. It's very technically very hard to make a weakened virus. So there are a couple in production, but they aren't made yet. And there are a couple inactivated virus vaccines. I think those are the ones you're seeing in China and Russia, but we don't have any in the US. So I wanted to give you two buzzwords to think about when you're listening on the news about how vaccines work. So the first one is immunogenicity. Immunogenicity basically means that they give you the vaccine, they draw your blood, and they look for your blood to have an immune response, which are antibodies. Antibodies are the part of the immune response in your body that helps protect you against seeing the live virus in the real world. So they give you the vaccine in your arm, they draw your blood, maybe like 10, 14 days after your second dose, and if your level goes up nice and high, they say,
Side effect of the vaccine is instant paralysis while giving presentations.
fast. You only just give 2 doses and wait 2 weeks and draw your blood. And you can do it pretty cheaply too, because all you have to do is draw the blood. But you cannot tell people that your vaccine protects you, because you're actually not getting true protection. You're getting a blood level of antibodies, which has not been necessarily shown to relate 100% to protection. So that's quite important to remember. The immunogenicity can hint at protection, but it's not the same as protection. Now, efficacy, on the other hand, is when you actually show that the vaccine works. So basically, you give the vaccine to someone, you say, hey, go out there and hang out in the world, go to the Super Bowl, go do some indoor dining at Red Lobster, and see if you get sick or not. And then if you show that the vaccine actually stops you from getting sick, that's what we call vaccine efficacy. Those studies, again, have to be a lot larger in size. They're a lot slower because you actually have to wait for people to get sick, and a lot more expensive because of the size and the duration. But at the end of the day, you can actually say that the vaccine, quote, works, actually stop you from getting sick. So I just wanted to give you a little bit of just details about that some more. So here's some examples. This is a little bit complicated to show, but let me get my little pointer out again. So on your left here, here's an example of immunogenicity from the Novavax trial. So Novavax, we gave 1 shot here, and we gave them another shot here. And at the beginning, we drew some blood, and the blood titer level was low. It was before the vaccine. People did not go out and get sick with coronavirus, so they had a very low titer. After 1 dose, and this is they use different levels. You can ignore the different lines. But after 1 dose, the titers were higher. Hey, look at that. The vaccine works. We got some titers going. It's only showing you that the vaccine creates antibodies. And after 2 doses, it was much higher. After 1 dose, it stayed the same. So look, this is basically saying we give the vaccine, we draw some blood, and we see antibody titers, and they go up. But what you don't have here, which is a key part, is what you're lacking is something called a correlate of protection. If you have a certain line in the sand, if you can say, look, If your antibody level gets above 10,000, like you have here, we know 100% that you are protected. Then this blood test alone can take the place of a very expensive, long efficacy trial. So again, you draw your blood if it goes from here to here, and we know that this level has been proven by the scientists to be associated with protection, then you can actually just do your study with a blood test. That's how flu vaccine is done. If I wanted to make my own flu vaccine, I wanted to call it Toback super flu vaccine, all I have to do is take it, give it to a bunch of people, draw their blood, and if my titer goes from here to here, I can get that flu vaccine approved. I don't need to take 30,000 people, give half vaccine, half placebo, and tell them to go out for 6 months and see if they get sick or not. So again, this is immunogenicity, blood levels, and we can't say anything until we have this correlate of protection. They are working very, very hard to establish this. And once they do, they hopefully can do these studies a lot faster. Now compare and contrast that with true efficacy. Here you're actually seeing people get sick. You know, each of these little lines here are people getting sick over time. So there's 1 sick person, 2 sick people, and this line here, this flat line, is the number of people that are getting sick that were vaccinated. So you're seeing very, very small numbers going up very, very slowly. So the number of people with the vaccine are actually not getting very sick. The number is quite small. It's less than that 0.5%. But those that actually got the placebo, 1, 2, 3, 4, 5, and getting sicker and sicker and sicker as time goes on. So this is actually showing these people are getting sick because they got placebo. These people are not getting sick because they got the vaccine, and that is what we call vaccine efficacy. The other thing about these trials, you might sometimes watch. Sometimes you hear it on the news. You say, this is a trial of 30,000 people was done, and they're doing an interim analysis, and so far with 100 patients, they've seen results. And you say to yourself, wait a minute, they vaccinated 30,000 people, and they look at 100, and you're telling me it works? How does that work? What does that mean for safety and efficacy? What that means is for the efficacy side, they're only looking at people as they actually get sick. So again, they count people getting sick, 1 sick person, 2 sick people, 3 sick people, and as they count, they know exactly how many they need to count to until they can get statistically enough to show that the vaccine works. And that number is actually quite small. It's actually in about the number of about 20 or 30, but that number it doesn't kind of pass the red face test. So actually people are looking at numbers more like 70, 100, or 150. So looking at 150 people to see as they get sick. And the reason that they do it this way is called an endpoint-driven trial. You actually can count people that are getting sick in real time and then do the analysis in real time. Other trials typically are done over a certain period of time. For example, a cancer trial. Cancer trial, you might say, okay, we're going to start at time 0. We're going to give half the people one regimen of chemo. We're going to give half the people some other chemo. And 12 months later, we're going to see who dies and who doesn't die. We didn't want to wait 12 months. We didn't want to wait 6 months or 8 months. We wanted to actually see in real time how fast people were getting sick. And again, the speed of this trial now depends on how fast people get sick. And how fast people get sick depends on 3 things. Number 1 is how big the trial is. So you vaccinate more people, you're going to get more people the opportunity to get sick. Number 2, how much virus is around. If there's tons of virus making people sick, you're more likely to get cases. And then counterintuitively, also the last one is how good the vaccine works. If half your people in your study are getting vaccine and they're not getting sick at all, you actually get a very slow accumulation of cases because half the people aren't getting sick at all because the vaccine works so well. So those are the 3 things that they did. But when you look at the safety of a big trial, 30,000 person trial, they actually look at everyone. So 15,000 people got placebo, 15,000 people got the vaccine. They look at, headache, fever, pain, swelling, and the entire population. But when it comes to efficacy, they only look at those that get sick. And they do that to cut down time without cutting any corners on the technique. So why do all the studies that you see on TV look about the same? They're all 30,000 people. They're all about 1-to-1. They all have similar design. It's not because we all copied each other. It's all because most of the studies you're hearing about on TV are the ones that are being funded by Operation Warp Speed, and Operation Warp Speed handed us a playbook. It was kind of nice. Actually, it was basically a cookbook for how to design these trials that was designed by the NIH. So they actually gave us the best practices for designing these studies. There's nothing magical about 30,000 people. They just thought that was a good size to get the number of cases quick enough. So the studies you see on TV are all very similar because they were all designed by a similar group at NIH, and they're all funded by the U.S. government. So the safety requirements for these studies. Basically, the FDA told us that this could be no different than any other vaccine trials. We're not going to cut any corners on safety. We're going to look at events after 7 days after each dose. So there's swelling, there's redness, there's the pain at the site, also systemic side effects, fever, body aches, joint aches, headache, things like that. So those were all looked at, as well as what we call unsolicited adverse events, which is basically anything. So if your fingernails turn black, that's an adverse event. If you got, I don't know, nail fungus afterward, that's an unsolicited adverse event. So any adverse event after 21 days after each dose was also looked at. In addition, they look at serious adverse events, so the ones that cause hospitalization or serious medical problem, as well as any medically attended event. So if I go and get a headache that's so bad I need to go to the ER, that's counted as a different level of an event because I need to go seek medical attention because of it. The FDA told us that the minimal number of people that we needed to evaluate initially to get the initial emergency use authorization was 6,000 of your 30,000 people. So half of them could be under 65, but half had to be older than 65. And each of these studies also have what we call predefined safety pausing rules. So if you saw X number of cases of this, or 3 heart attacks, or 2 strokes, they would cause that study to stop. And you actually heard that in the news a lot. There was a case of some neurological problem with the AZ vaccine, and that paused the study, and they had to review the cases in detail. The emergency use authorization is also a somewhat lower bar. So to get the EUA, it's not really a true approval, and so the bar for hitting the EUA is a little bit lower than it is for final approval. But everyone that gets an EUA needs to go forward to get that final approval. So it's not like it's a sneaky back door. If you get emergency use authorization, you must continue your trial until it has enough data to get approval or not, and at that point it will get full approval. And again, this is basically saying that you think that there's a good evidence that the vaccine is working correctly and that the safety profile is adequate. And once you have these two things, you can seek emergency use authorization. I just want to say one or two things about the variants. So here's just a little picture of the spike protein, and here's a little variant that they're worried about. It's called the E484. This is the one that caused a mutation on the surface and is allowing the virus to escape the antibodies that are being made from the current vaccines. So one thing I'll tell you is that there's not just a couple of variants out there. At any given time, there are thousands of variants. The coronavirus does a very poor job of copying itself. It makes mutations every single time it copies itself. It's only the rarest instance where one of those variants is so good, it's so much better than the other variants at doing its job, which is infecting people and replicating, does it become the dominant variant, and then it gets given a name, it's looked at and talked about, like the UK variant, South Africa variant, and the Brazil variant is one in Japan. So once they get enough of them that is taken over and it shows particular characteristics and they can talk about it, that's when they give it a name and they talk about it a bit more. The scary one is this 44, like I told you about, which is causing the ability to evade or escape the vaccine. They call it an escape mutation. And because of that, because of escape mutations, we're likely going to see two things. Number one is that all the major manufacturers are already making vaccines with these new variants. They're already underway. They're already making the vaccines. They're already doing the initial testing. And it's quite likely that in the future, now that we have multiple variants out there, with the original Wuhan, South Africa, who knows the other ones, there's probably going to be an opportunity to make a multivalent vaccine going forward to keep protecting people in perpetuity as the variants come up until we can get enough of the planet vaccinated that we reach herd immunity and actually shut this virus down. So we probably are going to see like the flu vaccine. The flu vaccine has 3 or 4 strains every year. You might be seeing multiple strains. You might see a Wuhan, South Africa, UK a vaccine come out in a year or two from now. I just want to show you what a variant looks like. So they can actually trace these variants over time. These different colors are basically just the different variants over time. And this is a little bit of data from the study that I conducted in the United Kingdom. We were extremely lucky that when we actually ran our trial, it was right at the exact same time that this new variant started creeping up and actually became the dominant variant within like 2 months. And because of that, about half the viruses in our study were the variant, another half were the routine. We would happen to be at the right place at the right time to actually get vaccine efficacy against the original strain and the variant because the timing was right. Somewhat similar thing happened to our study in South Africa, but it was a little bit later. We didn't actually catch it on the upswing. We actually caught quite farther into the upswing, where 90% of the cases that were accumulated in South Africa were already the South Africa variant. So we didn't actually have enough cases of the original to get a good vaccine efficacy against the original number as well as the variant. We must have had almost entirely variant. Although if we keep the trial going long enough, we might have enough of the original to give you a nice number against the original. So one of the reasons that Novavax has data against these variants is because we were actually just lucky. We're in the right place at the right time. And if you're thinking that these variants, the South Africa, the UK, are going to stay in South Africa and UK, I think I have a bridge I want to sell you because that's not the case. So they're already looking at the UK variants that's coming up, and it'll definitely become the US variant very soon. It was so much more efficient at spreading in the UK. It became the dominant strain within 8 to 10 weeks. We're going to see this pretty soon. And the whole reason that we're not talking about this more in the US is because we do a lousy job of testing. If we were testing 10% of the isolates that were tested everywhere in the US in sequencing, you would find out that the proportion that are the UK variant is going up. It's probably about 10, 20, 30% by now. We just don't know. At least I don't know. I haven't watched the news. I've been working too hard. I'm just in the news. And then what the funny thing is that now we actually have competing variants. We have the UK one that has a head start, which is actually very, very good at spreading more efficiently. And then we have the South African one, which is coming up from the East Coast, which is as good at spreading, but it's also good at infecting those that get vaccinated. So we are really at a race to see if we can vaccinate as many people as we can to really tamp down the levels of that South African variant. Because right now, South African variant can crash over a whole host of vaccinated people. You could give the South African variant to one person in a nursing home that got vaccinated 3 months ago, and it would wipe out the whole nursing home because it's a bad variant and it's evading the vaccine. I'm being a little dramatic. The efficacy of some of the vaccines were okay to the South African variant, but they weren't great. But the scary thing is it shows that the virus is smarter than us. So I put some numbers up here. Some of these things I couldn't put all the references on here. I usually like to put some references. But Matt specifically told me you wanted to know where we are with the data in terms of the major players and where are the efficacies. So some of these are up here, and this is I'll say my caveat. This is a mixture of some publications, some large studies, some press releases, so no actual publications yet, and also some data were based on very, very small numbers. In fact, I didn't even put our number up here because it was only based actually at the time on one case. So let me go through this for you. So Pfizer and Moderna were the first guys to come out. They got their vaccines, conditionally approved months ago. It was before some of the major variants were in place. So they actually have no variant data because they just weren't around. Now what they do have is they have immunogenicity. Remember I talked about the immunogenicity? You draw the blood and it goes up and you say, aha, I'm getting antibodies. They can show that there is an immune response to some of these variants, but it's a reduced immune response. That's one thing. But also, since the immune response right now is not correlated with protection, you don't know what that means. So if I say, look, when I get vaccinated, my immune response goes from here to here. Okay, what does that mean? Now, with the variant, it goes from here only up to here. Is that great? Is that low? Is that high? Is that sufficient? We don't know. So all we know about Pfizer and Moderna is they have no true vaccine efficacy against these variants yet because they haven't done studies where they're circulating. But they have some idea that they probably have some protection, but it's probably not as robust. But again, it's all just based on blood work. And then the other one here is they got very good numbers against severe disease. So they came out with their data. They had dozens of cases of severe disease, and they had pretty good efficacy against severe disease, which is great. Now, AZ and J&J are the next category. So these are now the the viral vector vaccines. The viral vector vaccines, one thing I didn't mention to you, which is kind of odd, is sometimes actually they put the mRNA that they want, the spike protein told you about, inside a virus, and then they give you the virus. Now, sometimes you've actually been exposed to this virus in your past, like adenovirus, and sometimes your immune system can actually fight off the vaccine because it's an inside a virus, and you might have protection. So one of the reasons that people are speculating that the Oxford and J&J vaccines aren't as efficacious, aren't as high, is that the human body actually is able to fight off the vaccine. So when they looked at the vaccine efficacy against the original strain, they were done at the time that there were variants. So they had to pull out the non-variants and calculate the efficacy. The numbers were quite low, or quite lower, 60-70%. Now, I wanted to put this in context. Anyone here get a flu vaccine? I get a flu vaccine every year. The average flu vaccine probably is about 50, 60, 70%. So when I say bad, I'm actually just saying it because the bar was set so high with these mRNA vaccines, 95%. This is actually an amazing, almost once in a lifetime vaccine efficacy. It's so rare to see such a great vaccine efficacy from a new vaccine. The shingles vaccine, high 90s. If you get 3 doses of hepatitis, you can get in the high 90s. So this is actually a really amazing result. So these results here actually aren't that bad. In fact, if you look at the initial success criteria, what did the FDA tell us was the target to aim for? It was actually 50%. They say if you can get 50% or greater, you're in good shape. So these guys are in good shape. They're just lower, 60-70%. The AZ vaccine, for some reason, actually had a little bit higher numbers against the UK variant. I don't know why that is. I think it might be just how the study was designed. The AZ guys had a lot of funny things going on with their trial. They actually had better efficacy when they messed up their dosing and they actually used a half dose by accident and actually had better results. They also looked at those people that got the vaccine instead of 4 weeks apart, they got much longer and they got better results there. So there's some funny ways that AZ has been looking at their data because it's not that great. The one thing that is for sure though is they had a small number that were tested against the South Africa variant, and it was not effective. So the vaccine efficacy was about 20%, but was not statistically significant. So you can't even say anything about 20%. They didn't have a large number of severe cases, but the severe cases were pretty good. So that's actually the one thing that's very reassuring about all the vaccines is that severe cases, being hospitalized, getting kidney injury, going to the ICU, having a very low oxygen saturation have all been pretty good. J&J, also lower on the side against the original. They don't have any vaccine efficacy against the UK because they haven't done studies in the UK, but they might eventually. They did studies in South Africa, and they got a number in the mid-50s, which again is similar to a flu vaccine, so not great. And their vaccine efficacy against severe was pretty good too, 85%. And then last is my vaccine here in the end, which is the Novavax. Our vaccine efficacy against the original strain, based on our interim analysis of our study in the UK, was in the same range as Pfizer and Moderna, so very high, mid-90s. I told you that we also were very fortunate to have a vaccine study done in the UK when the variant was available, where we could actually pull out the efficacy against the variant. It was actually still pretty good. It was in the mid-80s. Again, so it's pretty small numbers. Our final analysis coming out probably in a couple of weeks will have bigger numbers. Then the efficacy against South Africa was also low, very similar to J&J. And I think the bigger picture here is not that, my God, look how low the vaccine efficacy is. The bigger thing is this is a public health warning that some variants are escaping vaccines. So this is a warning to all the manufacturers to start making this as a vaccine, and also letting people know that it's possible that some of the variants can't escape the vaccine. So when the initial press releases came out and the news picked up as these numbers being low, I thought to myself, they're missing the point. The point isn't that the vaccine efficacy is lower. The point is, oh my goodness, there's escape variants out there. We need to start rethinking this whole process. And I didn't put down a number for severe because we had very few cases. I don't know, the guys in the UK probably take care of their patients better. We didn't have enough severe cases to give us a number, but I think we'll have more for our final. And I expect our number to be in line with the other vaccines. So good against the severe cases. So In conclusion, there are a large number of excellent vaccine candidates out there, and a large number of excellent vaccines, or not a large number, a handful of good vaccines. Their safety profiles are all pretty good. So you see some pain, some swelling, some redness, but overall we're seeing a very good safety profile for all the vaccines. Their efficacy profiles are all a little bit different. Some are very high, some are moderately high, but some also have attributes that are more interesting, like the J&J one. We had mentioned that was after a single dose. It's kind of nice to have efficacy after a single dose. But we also have other unusual things like the Moderna vaccine requiring ultra low temperatures, or the Pfizer even lower. The Novavax vaccine being stable in the refrigerator at 2 to 8 degrees Celsius, which is kind of nice too. So we have different profiles, but I think it's going to take a village of vaccines to really vaccinate this whole planet, because we have to vaccinate the whole planet if we want to take care of this virus. It just can't be the Upper West Side, the fortunate people. It's going to take the low and middle income countries that have to be vaccinated too. These vaccines can all get approved conditionally or with emergency use authorization, but they all need to go through rigorous final testing to get that final approval, and all vaccines will get that final approval if they submit for an emergency use. And then the last thing is the variants are out there. It's quite likely that we will be making a variant multi-valent vaccine in the future with some of the original strains, some of the new strains, and it might actually change every year. Until we actually get this vaccine, this virus under control, we actually might be looking at coronavirus becoming a seasonal vaccine like influenza, where it changes every year. So the flu vaccine, it's got 4 strains. They're named after the countries and the regions that they're found in. Quite likely the same thing will happen with coronavirus. And then in the future, we actually might even see combination vaccines. You actually might be able to get a flu coronavirus vaccine at one shot. So we don't really know what's coming, but I know a lot of people in my industry are thinking about this. So with that, I will take my final goofy Star Trek slide and take any questions you might have.
Thank you, Seth. There's some questions that came in via email before the session, so I'm going to hit you with 1 or 2 of those before we open up the mics for questions. And there's been some questions in the chat, but people can re-ask if they want, or I can go through the chat. The one that we got most often was the one that I think the news has been least clear on. Once someone has their 2 shots and they've waited there 2 or 3 weeks, for maximum efficacy, are they still potential carriers that can infect someone else and they're just not getting symptoms? Or are they not going to get it and they're not going to give it to any of their friends and loved ones?
Yeah, I think that the easiest answer is we don't know. I'll be honest that a lot of these studies were designed with no bells and whistles to look at very little beyond answering 2 questions. Is the vaccine safe? Is it effective? If you wanted to answer that question, what you would have done is you would have taken everyone that got sick and look at them, see how long they got sick for, and swab them maybe daily for a month to see how long does a sick person shed virus for. The other thing you would have done, though, is take a random sample of people that didn't get sick and swab them and try and find out the people that got vaccinated, maybe they did get sick and they're shedding the virus. So what we don't know is basically, the way they find out the vaccine works is I'm a vaccine recipient, I get sick, I say, hey, I'm sick, I call the site, they bring me in, they swab me, they see if I have coronavirus, and then at the end of the study, they unblind me and they say, did I get placebo or did I get vaccine? They don't take the people that aren't sick. If you're not sick, you're at home, do whatever. You're not coming in. No one is sick in a low-level carrier. To do that, it would have taken a lot more bells and whistles and a lot more work in that study to say we're going to randomly bring in 10% of the population, swab the crap out of them all the time, and see if they are low-level shedding. So some of the studies are looking at that a little bit, but they're really, that's going to probably have to be answered in what we call a post-licensure fashion. So after the studies the vaccines are licensed, there'll probably be more studies like that, and they'll probably actually use better technology like the Saliva Assay, where they can take people and have everyone spit in a tube every other day. And that way you can stay home. So Matt, you can be involved in a vaccine trial. You can say, hey, I feel fine. I would say, good, Matt, stay home, but spit in a tube every other day and mail it to me. And that way we can find out that Matt, who's feeling fine, hanging out at home, talking to his family, eating hot chilies with his brothers, is actually sick and shedding the virus, but he doesn't know it. Those studies haven't been done. a robust fashion yet.
Yeah. And is there any chance they'll find data like that out in contact tracing when if people are getting sick and the only one they've been around is vaccinated people?
They probably can. So there are epidemiology ways to do this. So it's possible. Yeah. In fact, I'm hoping that I get some egghead from UCLA will come up and want to do a large contact tracing or transmission study. College campuses, great place to do studies like that. Long-term care facilities, great place to do studies. So those are probably coming on the pike, and I think we'll have an answer to this question soon. But right now, we don't have a great answer.
Does anyone want to unmute and ask a question, or I can read one from the chat if you don't want to?
I have a question. Why doesn't the results of the first shot attack the second shot? It's seeing what it's looking for. Your antibodies are seeing what they're looking for in the second shot.
Well, that's a pretty good question. I will tell you that the first shot will start to give you an immune response and makes antibodies to the spike protein, right? So it depends a little bit on the vaccine, but in general, I think like the simplest way to answer your question is it does. So many people out there have gotten this COVID vaccine, right? Did the second shot hurt more than the first one? And why is that? It's because you're actually getting an immune response, and your immune response is basically your body is producing cytokines, it's producing interleukins and other things. But it's actually to some degree there's actually a little bit of the circulating antibodies making an immune response with the vaccine. So I think there is a little bit of that going on. It's not robust enough to actually shut down that second shot, but that's why it hurts a lot more. So I think you are seeing that to some extent. It's probably the easiest way to answer that.
Thank you.
Sure.
So Seth, an interesting question that came in on the chat from my buddy Marcus was, how do you go from EUA to full approval? Is that just finishing studies that were ongoing, or is there additional hurdles you have to go through?
Yeah, so typically these studies they do an interim analysis and they do a final analysis at a certain number. I can tell you like Pfizer and Moderna did the first cut at like 100, the second cut at 150, 180 cases. What they need to do is they need to then probably do a last cut of data looking at efficacy. They also will keep collecting safety data. So as these studies are going on, they're still collecting adverse events. So that they're 2 months after the second dose, 3 months after the second dose, 4 months. So at the time of submitting for final approval, they might have 6 months of data after the second dose. It's a more robust safety package. They have another maybe cut on the efficacy, and then they also have everything else that they might need, which might be additional manufacturing tests, test of stability, room temperature stability, other things like that. So there's lots of manufacturing quality stuff that need to be done in addition to looking at a larger safety profile. So it is a couple more things, and it's a lot of paperwork, it's a lot of analyses, and it takes a lot of time.
Any live questions before I go back to the chat questions?
Hey, Seth, this is Joey Carabetta. I got my second injection today from the Novavax trial that I'm in, and I'm very glad to see that blinded crossover looks like it's going to be approved. Is there any timeline? Is there any expectations of when that can happen?
Let's see. So let's see. I don't know if they made this public or yet or not. So I do know that they want to make that happen. They probably are going to collect a certain amount of blinded data first to get a good look at the vaccine efficacy compared to placebo. I don't know if that's going to be several months. I don't know if they're actually going to design it up front and say that we're going to do it at 4 or 6 or how many months. But they are going to have the opportunity to get everyone 2 doses of whatever they got first, then 2 doses of the opposite. So if you got 2 doses of placebo, you get 2 doses of real. If you got 2 doses of real, you get 2 doses of placebo. You won't know when. Exact timing, I don't think we've made public, they'll probably be several months into it after they've gotten enough of a signal from the placebo-controlled aspect of the trial.
All right. Thanks for your talk today. I really appreciate you joining us today.
Sure.
Yeah, I got the I think it got IRB approval for the blinded crossover. I don't. They need any more approval, do they? To do that?
I know that they've gotten feedback that it's good from the FDA. I actually don't know. I don't attend all the U.S. meetings. I'm the lead on the UK site, but I have enough of my own meetings. So I only get the wrap up. So I do know that they're getting feedback from the FDA. They're generally acceptive of the concept, and I don't know if they put it in the public domain what time they're going to do the crossover. But yeah, it's definitely going to happen. It's really the only good way. It's really it's the only way to ensure people are going to get treated the way they want to be treated in a vaccine trial, which is getting the opportunity to get vaccinated with the vaccine in their study. It's the only way to really reward people for all the efforts of being a volunteer.
Well, yeah, they don't want to keep people. I mean, people might drop out if they don't do it, right? Yeah. But it says so. They got IRB approval for that on the 13th of January. So I'm sorry, at the protocol level. Does that mean it's? I mean, if that's the protocol level, sounds like it's a done deal to do the blinded crossover.
Yeah, it might just be that they're getting final comments from the FDA. So the FDA is the U.S. regulatory body. The IRB is the
right.
That oversees the dynamics and the logistics of the trial. There are different groups. You need approval from both.
Okay.
But yeah, I mean, once you start going down the path of getting IRB approval, FDA is giving you the nod. Just a matter of time.
Yeah, and I think you might have already said this. The crossover won't happen until the emergency use authorization goes through.
I'm pretty sure they put that in the protocol as well. Yes.
Yeah.
So they need to show that the vaccine works first, because you can't compare the efficacy of the two arms, the early vaccinators versus the late vaccinators, until you get at least some efficacy with placebo. So yes.
Okay. Thanks.
Sure.
I have a question. I had COVID-19 in early December, and I was wondering, I'm a teacher, so I'm going to be eligible soon to have the vaccine. So I wanted to know, what is the sweet spot time to try to get the vaccine if you already have antibodies? Should I wait? Is it going to resist, sort of like what you described with some of the vaccines? Am I going to build? What's the sweet spot? Do you have any idea of that?
Yeah, I don't think there's a real scientific answer, but the best advice I can give you is that we do know that the immune response after COVID infection can be highly variable, and depending on the severity of your disease. So if you were sick in the ICU, you might get a higher titer. If you were moderate, you might get a moderate titer, and if you were less sick, you might get a lower titer. And so therefore, I would say that most vaccines on the market right now give you an antiviral response that is equivalent to or higher than those that were severely ill. So if it was me and I was mild to moderately ill with COVID, I would get the vaccine as soon as it was offered to me.
As soon as possible, or delay?
No, I would get as soon as I could, because I don't know how high my titer is. Again, it's a titer. I don't know how protective it is, but we don't know how protective. And we also don't know if natural infection to the Wuhan strain is going to give you any kind of cross protection. So we already know that for example, the J&J vaccine and the Novavax vaccine are at least 50 to 60% protective against the South African. We don't know if that's the case for natural infection. In fact, I would tell you that at least in the initial work that we did in South Africa, those that got sick with the original strain were not protected at all. So if it was me, again, I would get a vaccine and try and get that 50-60% protection against other variants that was absent in those that were naturally infected.
Okay, thank you. What are your thoughts on the antibody tests? Are they fairly effective?
Watch Seth.
What's that?
Don't say anything bad about Vitus. I'll come after you.
Oh, yeah. No, I was going to say it depends what your question is. A lot of people, we get a lot of people. It's kind of funny that they're trying to unblind themselves. They go in the trial and they say, "Hey, I want to find out if I'm in the placebo arm or the real arm, so I'm going to go get a COVID test," and they get the wrong test. It tests for the nucleocapsid. There's two. There's a couple of proteins on the surface. Our vaccine only has a response against the spike protein, but there are tests out there that test against other proteins like the nucleocapsid. So the most common ones, the ones that are on really big platforms that Matt's giving me a hard time about, like the Roche and Abbott, they test against the nucleocapsid. So if you just go out to a regular LabCorp, chances are you're going to get one of those tests, and you're going to go, darn it, it was negative. I guess I got the placebo. And in fact, you probably could have got the active one, but you just got the wrong test. So I would say it depends what your question is, why you're getting the test in the first place. Most of them are also not quantitative. They just say antibody yes or no. They won't say high titer, low titer, medium titer. And again, we don't know that what titer is protective. So if I say, hey, I got a COVID test, I had a high titer.
Yeah,
what does that mean? What does that mean? Is it protected? Until we have that correlate of protection, as soon as we have a titer that can say, my titer was 10,000, therefore I know 10,000 above is protective. An answer to that question is in my mind meaningless. It just says you have antibodies, which is nice to have, but it doesn't give you any more added benefit that you know you're protected.
I've got one too. So I've had the vaccine. I've had the Pfizer. Had my second dose about a week and a half ago. I didn't really get much of a reaction at all to either dose. I'm wondering, do people have different levels of protection? Can you tell by getting an antibody test whether you have good protection or not?
So we do know that people over 65 tend to have less side effects than those that are under 65, so it's not necessarily a bad thing. And again, an antibody test is not going to give you an answer about the quality. Most of them are qualitative. They just say antibody present, yes, no. It's black and white. It doesn't say high, medium, low. And even again, if it had a qualitative test and it said it was high, maybe you could feel better about that it was maybe a little bit better. But because you got the vaccine, we know it yes as true vaccine efficacy. But I don't think they've actually I don't think they tried to tease out subpopulations and say, oh, the people that got less side effects had a lower vaccine efficacy. The people that had bad side effects had higher. I don't think they've done that yet. Maybe they will, but I would say your best bet is knowing that you have a high efficacy vaccine, 95% on all comers, whether they were high risk, low risk, over 65, under 65, and just know that you got a good vaccine, and chances are you'll be in that same category.
Thank you.
Well, Seth, one of the other questions that came in the chat was St. Fauci said that single dose vaccine efficacy for COVID-19 survivors, just a single dose for people who had COVID, is something that he thought was worth investigating. Do you have any thoughts on if that's something that post-market is going to be done by any of the vaccine companies or should be done?
It will be done as a booster because everyone eventually want to get their original 2-dose regimen will get follow-up boosters as a single dose. So that will be done. But the other thing is almost in all vaccine trials, you think about this, if you vaccinate 30,000 people, what proportion of them are going to miss their second dose for some reason or another? Probably a handful, maybe 5, 10, 8%. So you'll be able to pull out those people and actually calculate a single dose efficacy. You can also calculate the efficacy between dose 1 and dose 2. So between those two populations, between dose 1 and dose 2, and the 5% that for some reason forgot about their second dose or were non-compliant, you can actually calculate a 1-dose efficacy for almost all your vaccines. And they'll be doing that when they do probably post-hoc analyses of the initial trials. So I imagine most vaccines will probably have a fair vaccine. I mean, if a vaccine is 95% effective, I can't imagine that single dose wouldn't be in the 70s to 80s. And I'm pretty sure Pfizer does have data on a single dose in the 70s. I think you're on mute.
Wrong button. Question that my brother asked me to ask you. So he got his first shot. He's in California, and he's like, "Well, if they tell me the first shot makes me 74% protected or whatever his number was, why does, how urgent is it to get a second shot? Is there a chance the first shot efficacy goes down if you don't get that second shot, or is it just to close the 20% gap?
Yeah, I would say, I mean, it's a kind of like a complicated question. It will bring it up to that gap, but we also do know that antibody titers are higher, that cell-mediated immunity, the other arm of the immune system, you got the antibodies, you got the cell-mediated, you get more cell-mediated protection with a second dose. I would walk over hot coals to get my second dose.
I told them to walk on hot coals regardless of whether they're giving doses.
Yeah.
So another question that was, when you're going to collect more data for a new variant, so if the other companies are adding the UK variant, or you're trying to add the South African variant, or any others that come out, how involved is the clinical trial for that? Is it like starting over a new clinical trial, or is it a smaller amount of patients and time just to add that level?
Flying in whatever until probably the summer. Well, they have they have upgraded the vaccine between now and then, so that we have one that covers the antibodies.
It will probably be in clinical trials. There might be some. I think it's likely that the fastest players out there will have some small initial clinical trials. They might also be doing booster studies for those. So I'll give that a maybe. Yeah, I mean, they're already manufacturing. I mean, like it took Moderna 45 days to make their first candidate. If they can beat that by a week, they can make their second one in 40 days. They can get that vaccine out in no time. We've already made ours, and ours is protein-based, which is a lot more complicated to do. So I would say there's a good probability that they'll be out there, about whether they'll be out in numbers, and they'll be in clinical trials. I just don't know about the numbers. Sure.
Seth, I have a question. What are your thoughts going forward in the future of the mRNA technology? Do you think that's likely to become the dominant player in vaccines in the future?
It seems to be pretty fantastic. I think that it is going to be a big player. In fact, I know Moderna is working on a whole bunch of other hard nuts to crack right now. So I think that they are going to use more mRNA for things like RSV vaccines, or other vaccines. They may even take some of the ones that have suboptimal results now and re-engineer them. It's really no reason why they couldn't make a better pertussis vaccine, or a better measles vaccine, or even hepatitis A, B, or C vaccine. So yeah, I think that there's going to be some strong futures. So I hope you're not asking about stock tips, but I think it's going to be great technology. It already has proven itself. It's the newest one, and I think people tend to be a little bit afraid of new technology. But I think in time, people will get over the idea that it's a piece of DNA that's going to somehow weasel its way into your genome and start causing you to sprout spike proteins in your body. Not going to happen.
So could they use it for flu vaccine, because you can manufacture it so much faster?
Sure. Yep. They probably could. In fact, flu vaccine is now being taken over by new recombinant technologies. In fact, I used to live in the town of Holly Springs, 35 miles south of here, which has a huge Sequoias plant. You can't see it because it's all hidden by the trees. It was built by the U.S. government. But that's one of the largest manufacturing plants that's not using egg-based technology. It's using recombinant technology. So it's right here in our backyard. So that's going to be that's the newest, best technology we have now. But there's really no reason why there couldn't be an mRNA influenza vaccine. Sure.
Good.
If there are no other questions, I'll ask one last question before we close up. Anyone? Bueller, going once? Okay, Seth, last time we saw each other in person, so for everyone to know, Seth has favorite viruses, and Seth's favorite virus back in February, and has been for years, is the flu virus. And I think SARS-CoV-2 was number 3 on your list at that point in time. So I'm wondering if the rankings have changed and what your favorite viruses are.
Yeah, I will say this. I did change my screensaver from influenza to SARS-CoV-2, so now I can stare my enemy in the face every single day. So it has taken this place on my screensaver. It was the one that was on my very first slide. But I'll still say I'm probably going to stick with the same ranking. Influenza was my first love. It's near and dear to my heart. I've got a couple of great books on influenza. It's got, even the name, influenza, it's Italian. I forgot the exact origin. I think they're thinking about it was some sort of influence coming from the gods making you sick. It's got great history. It's got some great science behind it. So that's still number one. I think, maybe coronavirus could have flip-flopped with RSV, but in general, still flu. Yep.
Cool. I'll make sure everybody at work knows that.
All right. I appreciate that.
No problem. So, Seth, on behalf of Triangle Free Thought, I want to thank you for taking time to come on. I know you just came back from vacation, and the last thing you want to do is jump into a presentation, but thank you so much for doing it for us.
Oh, great. Well, I want you guys to make sure you go out there and dispel any myths. We all need to be small vaccine advocates every chance we get. There's a lot of, bad thinking, wrong thinking out there. And I think the more people that are educated and can tell people about stuff, the better. And again, I've armed you with some vocabulary words so you can go out and watch that news and say, that's not real efficacy, that's immunogenicity. It's not quite as good. They needed a real study on that. We'll see. We'll wait 3 months to see what the studies look like. So thanks. I'm going to go probably do some more email.
Thanks so much, Seth.
All right. Bye-bye.
Thanks again.